We have Dr. Michael Lisanti with us with the University of Salford, and we have Dr. Weiping Yu, a physicist in the aerospace community. How you doing?
I'm doing great. So happy to see you, Dr. Lisanti.
Good to meet you. Good to meet you.
Yes.
So I want to give a little background because we've been following your work for a few years, so this is finally great to everybody see each other in person, or at least in video, right?
Well, I'll have to come visit you guys in the States sometime. I'm originally from New York.
Yeah. So what made you go to England?
Well, my wife is Italian, and we had lived together there in the States for many years, probably about 10 years, and she was a foreigner. So we wanted to be closer to Italy.
Yeah.
And I know England is not Italy, but it's close, so we can go back and forth to visit her family. And we wanted to maintain my daughter's ability to speak Italian.
Oh, okay.
So, and a lot of this actually started from my daughter. I don't know if you remember.
Yeah.
So
she's the one that gave you the brilliant insight, right? That changed the world.
exactly. It was her idea to use an antibiotic when she was 8. Now she's 14. So 6 years later, we've done a clinical trial for the doxycycline in Italy. That worked.
Was that on breast cancer patients, right?
That was on breast cancer patients, yeah. And now we developed also a triple combination that includes azithromycin. And we think that also azithromycin, because of its senolytic activity and the fact that cancer cells originate from senescent cells, would also give us a way to do prevention. So we're going to do a second trial, which is conditionally approved. It's approved locally. Now we need the national approval in Italy. But we'll roll it out, I guess, once this pandemic is resolved. But it's a triple combination of azithromycin, doxycycline, and vitamin C.
Yeah, that's very interesting. How about berberine? You had used that as a combo too. Do you like vitamin C better?
Well, I think it depends on the mechanism. Berberine is another mitochondrial inhibitor, but the issue with vitamin C is that most people don't realize that it's actually a pro-oxidant. And what we're using it for in this case is a way to stimulate mitochondrial biogenesis at the same time in the cancer stem cells. And vitamin C actually is targeted with a very high efficiency to mitochondria. And at the same time, if we give the doxycycline and the vitamin and the azithromycin, that inhibits the mitochondria, their ability to make essential proteins that are part of the OXPHOS complex. So it's a way to accentuate their activity, because if you turn on mitochondrial biogenesis, at the same time you inhibit the production of essential proteins, you'll make a lot of mitochondria that are not as effective, and you'll ultimately starve the cancer cells to death, the cancer stem cells.
Wow.
It's a way so we can dial down the concentration of vitamin C, with a little bit of vitamin C, we can dial down the concentration of azithromycin and doxycycline, so it's submicrobial level. So you could avoid the antibiotic resistance problem by using the vitamin C and using lower concentrations of the antibiotics.
Right. That's interesting. So you don't have to use as much antibiotics if you've got the vitamin C, right?
Right. We could use, instead of using 25 or 50 micromolar of doxycycline, we could use just 1 micromolar.
Wow.
And same thing for the azithromycin. So we could use vanishingly small quantities of the antibiotics together with 500 milligrams of vitamin C in the trial.
So it's like a delivery mechanism, the vitamin C, in some sense.
It's a way to turn on the mitochondrial biogenesis at the same time to inhibit potentially the function of the mitochondria.
Okay, I see.
So that's our triple combination. The doxycycline by itself was actually quite effective. It had nearly a 90% response rate, reducing the cancer stem cells between 17 and 67% in our small trial. So we wanted to make it better.
How about azithromycin? Have you tried that by itself?
We haven't tried that yet in a trial, but it is effective in our assay system. It does inhibit the cancer stem cells as well. And we do see it has a strong effect on the senescent cells, removing them with an efficiency close to 97%. So we think that it's a sort of a killing two birds with one stone, as they say, because you can remove the senescent cells, and then you can also kill the cancer stem cells.
Right.
And so you can also reduce your risk, because the number of senescent cells, it's sort of like kind of like statistics. If you have more if you have more senescent cells, then you're more likely to develop one that turns into a cancer cell.
Right.
So if you reduce the number of senescent cells, you can reduce your risk potentially for developing cancer.
Yeah, and other aging-related diseases, right?
Exactly. So the idea is to use a senolytic, because the problem is, if you keep in mind that the senescent cells are the precursors for cancer cells, you wouldn't necessarily want to revive them. Some other people in the field believe that it would be better to revive the senescent cells, but we think it's better, less risky to get rid of them, because you wouldn't want to drive them into a cancerous state. So it probably would be less risky to eliminate them. And then this could also potentially allow the normal cells to regenerate. For example, the close relative of azithromycin is roxithromycin, and that's used actually for restoring hair restoration for
Right.
male pattern baldness. And that one also is a senolytic. So we're thinking what's going on there is that it's removing the senescent cells in the scalp,
Uh-huh.
allowing the normal stem cells to then flourish and then to regrow hair. So,
Right.
so wouldn't that be nice? You can prevent aging and cancer and grow your hair.
I like that. You can just get your nice luscious hair like Fabio, right?
For Michael. Okay, so since CPAC, mycin, is one of the antibiotics you use to cure cancer and to also for anti-aging. Does cancer cell have anything related to senescent cell?
Yeah, so we think that the senescent cells are the precursors for the cancer stem cells, and other people have suggested a similar idea. And what we saw is that when we isolated the cancer stem cells, that they still had senescence markers. So sort of like a kind of fossil record of what they were before. So we think that what's going on is that they're breaking out of senescence by potentially increasing their ability to produce energy. So we think that senescent cells are low energy and cancer stem cells are high energy. So they may be using the mitochondria and antioxidant capacity to escape from senescence.
Okay. What is the mechanism in your mind how this antibiotics works with the senescent cell?
Right. Unfortunately, we have no idea. And the problem is that we know that azithromycin acts as an inhibitor of mitochondrial protein translation as a side effect. But we don't know why it's selective for senescent cells. And we compared with doxycycline, which is another antibiotic that we see inhibits cancer stem cells and also inhibits mitochondrial protein translation. But doxycycline did not behave as a senolytic drug, although some people believe it still has anti-aging properties. So we don't know what's special about azithromycin. I can say that it is very selective, though, because we compared it to erythromycin, which is the parent and founding member of the class of drugs. And erythromycin, which also has that anti-mitochondrial activity, had no effect on senescent cells. It had no effect on as being a senolytic. So we think there's a big difference here, and it could be that because the azithromycin induces autophagy, that it may be an explanation for its senolytic activity, because we think that autophagy and senescence are a continuum. You probably start off with autophagy, and then something goes wrong, and the cells are already filled with lysosomes and with autophagosomes, and there are very dangerous enzymes in the lysosomes called cathepsins. And if those lysosomes become leaky, it's been shown by other groups that they can leak out into the cytoplasm, and they can also cleave the sirtuins, which may put the cells then into senescence. So we think that maybe senescence actually starts with autophagy, and then it's a progression. So you could think of senescence as autophagy gone wrong.
I have another question about, do you know why those virus has this crown looks like a shape?
The macrolide, right? Is that what they, or the antibiotics have the similar shape too, right? A macrolide, that macrolide antibiotic.
I guess there are two separate questions. I don't know why the virus has that particular shape, but we think that it is maybe something about the macrolide ring that is important, because roxithromycin also has a very similar structure
That's
to
wonderful.
azithromycin, but there are big differences with erythromycin. So we don't know what it is in particular about azithromycin chemically, but we are working on that.
And doxycycline, is that not a macrolide?
No, it's a separate class. It's a class of the tetracyclines. So we think that the tetracyclines will behave similarly, but doxycycline is one of the latest tetracyclines. It was actually first FDA approved in 1967, so 50 years ago. And it's only, I think there's only one other newer tetracycline, which is called tigelocycline, and that was made in case any resistance developed to doxycycline, but it doesn't look like there's been much resistance because no one's really ever heard of tigelocycline. So, and doxycycline is actually the #1 drug used worldwide for infectious disease. So, and people take it for 6 months at a time without many side effects. It's most often used for acne. So there are millions of people on doxycycline for acne worldwide.
Now, is there any, have you been able to observe any difference in the mechanism of how doxycycline attacks the senescent cells or these cancer stem cells versus the azithromycin and the other macrolide families that you've observed?
Yeah, so doxycycline doesn't appear to behave as a senolytic, but it does have other anti-aging properties because it does inhibit the mitochondrial protein translation, which appears to increase longevity. And also, there's a relationship here with the SASP. So, and also this ties into why we think these drugs may be relevant for the COVID-19 treatment. It's because they have a side effect. The side effect of doxycycline and azithromycin that they share is that they inhibit host protein synthesis. Now you need protein synthesis for two things that are relevant here. One is the SASP, because in order to make cytokines, you need protein synthesis.
Mm-hmm.
So if you use an inhibitor of protein synthesis like doxycycline or azithromycin or rapamycin, they'll all shut down protein synthesis and inhibit the SASP. So you won't get as much IL-6. So they have an anti-inflammatory effect.
That might be the mechanism. Right?
What's that?
Oh, that might be one of the mechanisms. So
Yes, exactly.
Stop the virus propagation.
Right. The other thing is that virus propagation requires protein synthesis.
That's right.
Viruses need to synthesize their proteins to make new viruses. So if you shut down both the SASP and viral replication, that may be how the azithromycin is positively benefiting in the clinical trials the patients who have been, where the viral titers have been brought to zero. There was another trial which was just published online where the French group replicated their results. Originally, they did it in 6 patients. Now they've done the combination of hydroxychloroquine and azithromycin in a cohort of 80 new patients, where most patients behave similarly, and they were able to eliminate the viral particles.
Yeah, Dr. Lisanti. I have one interpretation from physics point of view about why the shape of this coronavirus is a crown this spike look like. You know, if you play physics, if you have a magnet inside of a ferromagnetic field, magnetic fluids, you will see those spikes all over around. So what I see when there's a crown-like virus, itself is a highly magnetic charged cell. So that's why I have those spikes. Those spikes, why they are not connected, those spikes are like poles, opposite. So that's why it's all spherical shape or all around. So so that's why it's because it's highly charged. So that's why it's so dangerous. It can, you know, because every cell has its magnetic property too. So that's high charged receptors can penetrate normal cells. And you mentioned about adult cells and
Senescence, yeah.
Yes, it is.
Yeah, those cells, because it's older cell, it could be imperfect. So the binding, the magnetic force, binding force may be not that strong. So that's why easier to be infected. That's what
Yeah, that's an intriguing idea. But I'm really not a physicist, so I can't comment on.
Yeah.
But I think, you know, obviously there's a lot to the problem, and we're not seeing all the parts of the elephant. So we have to keep, you know, take everything in consideration. And I think we'll need more clinical trials. But I think in the United States also there are doctors who are able to prescribe, because there is something called off-label prescriptions, and 25% of all prescriptions in the United States are considered off-label. That just means that the drug is FDA approved for one indication, but it's prescribed for another indication. So there is flexibility in the system, and that already exists. So the FDA does not necessarily have to approve these drugs for coronavirus therapy, because it already has flexibility in the system. It's at the discretion of the practicing physician whether or not they want to write that prescription. So that flexibility allows physicians in the United States to write prescriptions for drugs that are off-label.
Oh, okay. Professor, I observed another thing from your paper. You mentioned about this chloroquine.
Yes.
Has this called, you use the word called alkalines.
Yes, alkalinization to make the pH
pH
not acidic.
You know what I find that this is very, very similar to Chinese herbal medicine in Wuhan. So they treat those patients. You know, when they round up people in those huge stadium, and there's no particular medication work. What they do, they use a Chinese medicine, those herbs, and comes each person has a bowl of those soup, you know, cooked soup, and so that's how make people think, oh, I get treated instead of I'm just resting, right? And it has amazing effect, 97% of effect with this kind of Chinese herbal medicine. So I was thinking about it. You know what? These Chinese herbal medicine are more like alkaline type of things. But the sickness, the virus itself, create an acidic type of sick saliva in your lungs. So that's why people has a dry cough, because the saliva inside of the lung liquids inside, these are so sticky. So what the Chinese urban medicine do? They neutralize them. Like alkaline neutralize this is called acidic symptom and create salt and water. So that's why they can cough it out. So I'm thinking about the use antibiotics, the way you use include CPAC and chloroquine has similar effect. Basically,
right. Only that they also have anti-inflammatory properties.
Yes,
I
yes.
think what's going on is that what's killing the patients is the inflammation and the fibrosis.
Yes.
So if the lungs become fibrotic,
Yes.
then the patients can't respire. They can't expend their lungs. So,
yes.
But the cells that cause the fibrosis are the myofibroblasts. And the myofibroblasts are considered to be senescent cells. And there's a huge lesson to be learned from this from patients with cystic fibrosis. So patients with cystic fibrosis have a defective gene, the chloride transporter, the CFTR. It's actually one of the most common genetic diseases in humans, cystic fibrosis. And these patients die from lung fibrosis. And by accident, they discovered that if they give these patients azithromycin, they're fine. They extend their lifespan for quite some time. So they thought they were targeting a bacterial infection, but if you look at the literature closely, what they're doing is they're actually preventing fibrosis. So we think the mechanism, by the way this works, is that the cells that are causing the fibrosis are the senescent cells, and so they're being removed. And that would explain why the azithromycin is so effective, because it's a senolytic. Because one way to think about cystic fibrosis is sort of like a kind of rapidly aging disease like progeria, where your lungs are exposed to a tremendous amount of inflammation and fibrosis. And recently I found another study that was published in 2017 where they took patients with an untreatable disease called idiopathic pulmonary fibrosis. And they treated them with azithromycin because of the studies for many years on patients with cystic fibrosis. And they saw that they could dramatically increase their survival just with azithromycin. So again, this speaks to the antifibrotic property of the antibiotic through the elimination of senescent cells. And another thing is that aging is associated with fibrosis. One of the things that goes up as we age is fibrosis. So fibrosis is another hallmark of aging. So that could explain also the lethality. Go ahead.
Can you tell folks what fibrosis is for those who are not familiar with the term?
Sure. So fibroblasts, which are a kind of generic cell in your body, produce proteins like collagen that are very stiff and inflexible. So what happens is during an injury, the fibroblasts try to repair the injury by making more collagen, and this leads to scarring. So for example, if you cut yourself on your skin and you have a scar, that develops a keloid, which is mainly the deposition of collagen. So what the azithromycin would do is it would remove the myofibroblasts, which are the senescent cells, and you wouldn't get the scar. But think about that on the inside of your body, in the lungs.
Right.
You're developing all this scar tissue, which is inflexible. It's just like plugging the holes in a dam, but it doesn't have the same function as the normal cells. So you're replacing the normal cells with scar tissue, which is essentially non-functional. So then the lung becomes inflexible. It can't expand and contract. And so then you can't breathe.
Right.
So that's, again, what kills the patients with idiopathic fibrosis. It's what kills the patients with cystic fibrosis. And I think that's what's killing also the patients with COVID-19, because of the inflammation is driving that process. The inflammation is really the SASP, which is driving this this process of fibrosis and scarring.
So if that's correct, then that means that potentially azithromycin could be a good preventative or prophylactic, right? Or would we do?
But that's what I'm thinking, and obviously we need to trial that in clinical trials, but it explains the great success of the French group in combining. They didn't do azithromycin alone, I don't know why, but they did first the chloroquine, and they showed some benefit with the chloroquine, the hydroxychloroquine. It's just a derivative of it. And but
that's
then,
not an antibiotic. It's a totally different class of drugs, right?
well, it is used for malaria treatment and prophylaxis. There's an interesting story behind that too, because it all started with quinine, which is the original precursor, and so eventually quinine was modified and developed into what we have today, which is chloroquine and hydroxychloroquine. But quinine is also a flavoring, and it's what's present in tonic water.
Right.
So originally people started drinking tonic water to prevent malaria.
Yeah, like the British Empire, right, with that whole
Exactly.
thing.
Exactly. So I don't know if there's enough quinine in tonic water, but
I drank one yesterday just in case.
just in case. Okay. So, but I think the French saw this great synergy with the azithromycin, but we don't know if that's due to azithromycin alone, or if it's due to the synergy between the hydroxychloroquine and the azithromycin, because they didn't do azithromycin alone. So I think they would have to do also potentially azithromycin alone. That's not a criticism.
Right.
It just means that we still have a lot of work to do.
Now, this is interesting to me. Do you think that what I mean, if thinking out loud about, there's a lot of people who are doing this combination. They're imitating this because they're seeing that famous study in France. I just saw an interview with Dr. Vladimir Zelenko in New York. Have you seen him? And he's a doctor who he's got 100% success using the cocktail of hydroxychloroquine in combination with azithromycin and the zinc sulfate. And he's treating a lot of folks in New York in his practice. I think they said he's done 699 patients of coronavirus he's been treating with this with a lot of success. So what would you say to folks who are saying, well, why should we eliminate one of these three? Why should we eliminate two?
I mean,
right.
Now when there's a crisis, I think you
right.
can. But maybe in the future we can tease that out.
Right.
Because, some of the drugs have serious side effects. I believe the side effects for hydroxychloroquine are much worse than the side effects for azithromycin. But again, we want to minimize side effects.
Right.
So if you don't need to take something, it'd be better to know now than to continue taking both.
So it's possible that azithromycin alone could do the same level of effect that the 3 combo is doing with or even the 2 combo of hydroxychloroquine and azithromycin. Is it possible?
Right. It's also possible that doxycycline may be sufficient as well, because doxycycline inhibits viral replication. Doxycycline inhibits the production of IL-6. So there are papers published on, I'm forgetting the name of the virus, but there's a virus where they tested it. It's not Zika virus, it's dengue. So they used doxycycline as a prophylaxis and as a treatment for dengue virus. And they also showed in patients that were infected with dengue that it reduced dramatically the IL-6 production. So, and we know that doxycycline is also an anti-aging drug. It's been used in C. elegans in published studies to nearly double the lifespan of the C. elegans. So that would fit together too, because it's anti-aging in the sense that it's inhibiting the production of the cytokines like IL-6, which are driving the senescence-associated secretory phenotype.
The cytokines is the inflammatory response of the immune system that ends up killing these folks that get this lung disease from the COVID-19, right? That's usually what's getting them.
Exactly. So it could be the inflammation which leads to the fibrosis is the critical factor. So rapamycin, azithromycin, and doxycycline all share that property of being protein synthesis inhibitors. So they all are known to somehow affect and reduce the SASP, and then therefore reduce also IL-6 production.
Is it possible that they could work synergistically, doxycycline and one of the azithromycin or the related one?
The doxycycline, yeah. They all inhibit by a similar mechanism, inhibiting protein synthesis. So I think you'd only need one of them.
Okay.
Obviously, you don't want to, because every time you take another drug, you add the potential for additional side effects.
Right.
So you want to minimize the number of drugs you're taking. But I think with any one of those drugs, you increase the inhibition of protein synthesis, and then you can potentially reduce both the SASP, IL-6 production, and also reduce viral replication.
Now,
you want something that is inhibiting viral replication to reduce viral titers. So then the patients are not infectious, and also they don't continue to infect themselves.
Right.
So reduce the course of the illness to prevent the fibrosis.
Now, you had mentioned earlier when you were talking about your trial with using the doxycycline and azithromycin for cancer, right? You said vitamin C could enhance that. Would this work here, or this would not be something to try here?
No, I don't think you would need the vitamin C in this case, but you could use more than one inhibitor of protein synthesis to get a better effect. You could potentially dial down the concentrations of the two antibiotics to achieve something that was submicrobial.
Interesting. So
I think there's a lot of potential here, and we just need, and there's also a lot of patients that are in need, so they could easily set up clinical trials to test, and I'm sure the patients also would be willing. Obviously, they have to sign on for a trial. So there's a big opportunity here to test these drugs. And the only thing that's approved now, I think, in the United States, I just saw an article where they approved, because of the emergency, the FDA approved hydroxychloroquine, but they didn't approve the azithromycin. That doesn't mean you still can't get it off-label with an individual prescription, but they didn't approve the combination, which appears to be so much more effective. I don't know why. I guess it's a matter of time for them to realize that they just have to read the literature.
Well, from what I've understood, what a lot of doctors are interpreting azithromycin's role in this combo that they're finding, and it's not just France. I've seen South Korea and some other places have been doing this hydroxychloroquine azithromycin combination, from what I've read online, but they seem to interpret the azithromycin's role as trying to prevent secondary bacterial infections. It's not, they don't see the antiviral property of what you've described here, right? I mean, they don't see that. And I think they kind of...
Azithromycin, they just need to read the literature, because azithromycin has been shown to inhibit the replication of Zika virus and Ebola virus.
Right.
So they actually use azithromycin for these purposes, as far as I know. And, the antiviral, I think it's a misunderstanding among the medical community that people say that antibiotics are not good for targeting viruses. But if they understood the principle that viruses require protein synthesis for replication, then it becomes obvious that you could use an antibiotic that inhibits protein synthesis. It's not, so antibiotics all have side effects, and you just have to select the ones that have the inhibition of protein synthesis as a side effect. So we just need to broaden our definition of what antibiotics could be used for with the knowledge of the mechanism by which they're acting. Also, there are papers published with rapamycin showing that it inhibits HIV replication. Right. Why isn't rapamycin used for HIV patients? And I don't know.
This information needs to get out there, because I remember when recently when President Trump pushed very vocally azithromycin and hydroxychloroquine. A lot of media pushback, including from medical community, said this is absurd. There's no evidence that this antibiotic has any antiviral properties. And not to make it political, but that is something that has been widely touted, that this is an absurd idea that this antibiotic could be used on a virus like this. I've seen that touted all over the media.
Well, the people who said that should read the literature first before they make claims that azithromycin has no antiviral activity. They could just do a PubMed search or a Google search. Could take a few seconds. You just have to search azithromycin and viral replication, and you'll see there are papers published showing that it inhibits viral replication. It's a no-brainer.
Right.
Yeah, I agree.
Yes, you have to think before you speak.
Right.
You need to check your facts before you speak.
That's right. I believe this is a very important fact. I always know this. Have a virus infection, actually antibiotics works. Well, since 30 years ago, I came to the United States. The doctor always says, no, antibiotic does not work with a virus. I know the fact. With my family, my kids sick, they said, oh, this is a viral infection, but antibiotics does work. So I believe that's very, very important. I have another question. So Professor Lisanti, do you believe that the two types of antibiotics, they are actually strengthen the cure to virus instead of canceling each other?
I think it's possible. But I wanted to return to the virus and antibiotics idea for 1 second.
Yes.
So you can also find online that there's evidence that erythromycin is suggested, not azithromycin, but erythromycin, and also other antiviral drugs, other antibiotics are suggested for treatment of herpes virus. So for example, people use erythromycin for herpes viral outbreak. Why is that? Because it also inhibits protein synthesis. And people also use the supplement lysine, which is available commercially, to inhibit herpes virus as well. I've seen through my reading of the literature that lysine inhibits protein synthesis as well. That's sort of a kind of contradiction because it's an amino acid which is required for protein synthesis. But what it's doing is, my understanding is that it's inhibiting the uptake of another amino acid in the gut, arginine, and so therefore it inhibits protein synthesis. So there's a lot of data in the literature to suggest that inhibiting protein synthesis will block virus replication. And I also saw a patent application from many years ago where they used cyclohexamide, which is it's not a drug that's FDA approved, but it's mechanistically, its only known use is to inhibit protein synthesis. And they showed that that could be used I think in a small clinical study because it's topical. I think they showed that that could inhibit plantar warts. So they could cure a plantar wart with cyclohexamide. So again, it doesn't matter. So these are treatments that are not virus specific. So we're talking about ways of inhibiting viral replication that it's independent of the virus. So it could be used for any virus. So you could, as long as you inhibit protein synthesis, you'll inhibit viral replication. So you don't have to necessarily say, oh, well, we did it for, Zika virus and it's specific for Zika virus or HIV or Ebola. But if you inhibit protein synthesis, it should inhibit the viral replication for most viruses because you're inhibiting a process by turning off the ability of the virus to replicate.
Now, is there any other team that is making these same observations as your team? That's seeing the pattern here, that's seeing the big pattern here.
I don't know because unfortunately our university closed last week because of the virus outbreak. So at the moment, myself and my team are working from home. So we're writing up papers and thinking about this problem of the coronavirus as an aside. But I think that's important because, you know, major breakthroughs in science are often achieved by people who are not part of the field.
Right.
I'm not a virologist, I'm not part of the coronavirus field, but because of my broad interest in aging, it caught my attention that there was an association with advanced chronological age, and also because of our work with azithromycin. So it doesn't take long to read the literature and find this relationship between protein synthesis, viral replication, and inflammation that people are not talking about. So I think we need to
It's
talk about
huge. Yeah.
because it could reduce aging, could reduce cancer, and could reduce infectious disease, all with the same approach.
And they're
Just
all generic antibiotics. You know, I love
they're
that.
all generic antibiotics that cost almost nothing.
Unfortunately, I think there's a little bit of implicit bias in the way that we think of solutions because of the fact that they're generic antibiotics. They're so cheap. We always want to believe we have a bias. I don't know what the word is, maybe complex solution bias. We always want to, oh, if it's a new thing, we have to create this complex new drug and all this stuff. It can't be something that we use every day. I mean, to tell what you've done is so tremendous because, I mean, to tell the average person, and of course, it's a long ways away from being able to get to that point. But to suggest to the average person that an ordinary generic antibiotic like doxycycline or azithromycin could be used to help with cancer, you know, potentially down the road when you bear out more research, that's such a dramatic thought for most people, because most people think hundreds of billions of dollars for something like cancer. No, whatever the solution is for cancer, it's going to be a very complex, very advanced drug that will be patented for till kingdom comes, so to speak.
The good thing about this approach is that also it reduces the time from discovery to direct application, because most drugs, if you develop a new drug, it costs $1 billion and it takes 10 to 15 years. But if you take an existing FDA approved drug, it's already been through phase 1 clinical trials, so the safety has already been proved. So it goes directly then into a phase 2 clinical trial. So it saves all this time and money, and it still might even be more effective than a new drug.
Yeah. I think with the promise that you are showing as a potential for what can be done with these azithromycin and doxycycline and so forth, that, this is just kind of an aside point, but we've thought about, we need to manufacture these antibiotics in America if they need to have them in America, because they're talking about supply chain issues. You're talking about some countries blocking some of the export of their antibiotics and so forth. This is something that if Americans really want to be interested in, something that could not only help the COVID-19, but other aging-related diseases down the road that have been plaguing us for a long time.
Right. You could imagine it could be used for all the aging-associated diseases. And azithromycin, I think, came off patent in 2017. So the price of a generic would also be relatively inexpensive. But doxycycline, I think it only costs about 10 cents a day for 200 milligrams. Usually it's formulated as 100 milligrams per capsule. So the cost, I think, of a capsule is about 5 to 10 cents.
Wow.
I have a question. So what's the fundamental, what's the key difference between virus and the bacteria in your mind?
Well, there's an interesting, so a virus is just a piece of DNA or RNA wrapped in a protein coat. Many viruses also have an envelope. And so they just have the genetic material to reproduce themselves. So there's nothing really that complicated about them in the end. But bacteria are interesting because there's a huge relationship evolutionarily between mitochondria and bacteria. So about 1.5 billion years ago, bacteria snuck into cells and they became mitochondria. So that link is important for our anti-cancer work, because we think that the cancer cells need more energy than normal cells. And the way they do that is by using their mitochondria to produce more energy. So we can use the side effect and the evolutionary relationship between mitochondria and bacteria to fight cancer. You can use antibiotics because of their mitochondrial side effect. You can use that to kill the cancer stem cells. So the cancer stem cells need to make new mitochondria for them to propagate. So potentially you could reduce, so cancer stem cells are thought to be responsible for recurrence, metastasis, and drug resistance. So then you could use drugs that target the mitochondria to reduce all of that. And cancer patients actually die, 90% of them die from the metastasis. So wouldn't it be wonderful if we could use mitochondrial inhibitors like antibiotics to prevent the metastasis?
Wow.
It would be cheap and effective. So we need to do further studies, but this whole relationship, evolutionary relationship between mitochondria and bacteria deserves to be explored. For many years, more than 20 years, people ignored the idea of using mitochondrial inhibitors because they would say, oh, you're giving the patient cyanide. But we know that doxycycline is a mitochondrial inhibitor, and it's been FDA approved for over 50 years, since 1967. It's the #1 drug prescribed worldwide. So
does it inhibit the mitochondria of the cancer cells only, or healthy cells too, the doxycycline?
Well, we see a propensity that it's more specific for cancer stem cells. But we're not the only ones who have done this. There was a whole, the story actually started in Italy in the early 2000s. And there was a patient who had a canary. This all sounds very silly, but it's all published in the literature. So there was a woman with a canary, and she developed a lymphoma of the eye. And the doctor thought, oh, this is in Italy. They thought, maybe there's a relationship here. Maybe the bacteria from the canary infected the woman's eye and led to the eye cancer. This is called a MALT, M-A-L-T, lymphoma. So they gave the woman doxycycline and she was cured. So they did a whole bunch of trials in Italy, and this has been reproduced in other countries. And what they found is that if they give patients doxycycline with MALT lymphoma, they can get rid of the lymphoma. Also, patients that were resistant to conventional drug therapy, when they gave them doxycycline, 50% of them, they could get rid of the MALT lymphoma. So now I believe doxycycline is the second line treatment for MALT lymphoma. But when they checked to see if there was any relationship with bacterial positivity and efficacy, there was none. So there were patients that were completely bacterial negative, but they still had a complete response to doxycycline. So this means that the original idea was to treat cancer as an infectious disease, and it worked, but actually there's no bacteria there, or it has no correlation with the outcome. So what that means is it must be something else. We believe that is because it's targeting the cancer stem cells. And there was a similar trial in China for lung cancer. And what they showed is they could nearly double survival in patients with lung cancer by using azithromycin. Again, they thought they were targeting a bacterial infection in the lungs, but we think it's probably the cancer stem cells.
Wow. So does this
all published? It's all in the literature.
All these potential pathways, would this lead us to believe that perhaps some of these, again, upon a lot more studies, could treat the cancer completely in and of themselves without having to rely on the more invasive procedures of surgery, chemo, radiation? If you can, would it leave the tumors intact and not break it all apart, or how far can you go with these pathways potentially?
Well, the issue is that you can't do, legally speaking, you can't do a trial unless you do it as an add-on to the standard of care,
right?
Practically speaking. So what you'd have to do is you'd have to do whatever the standard of care is, plus doxycycline or plus azithromycin. to see. And so it could be used to fight the biggest problem, which I was mentioning, is the recurrence, metastasis, and drug resistance. So you could still use all the conventional methods of surgery, radiation, and chemotherapy, but then you could do a kind of protocol potentially of preventative medicine, because what you want to do is you want to prevent metastasis.
Mm-hmm.
If you can, that would be the holy grail of cancer research and cancer therapy, is to prevent metastasis. So if you could use these inexpensive drugs to prevent metastasis, then you would have a lasting cancer cure, because that's why patients fail conventional therapy, because of metastasis.
Right. And I know legally speaking, that's where we are with the standard of care. I'm just saying, hypothetically, is there a point in the future that you could actually use these as the standard of care? I know this is a big touchy subject for people, but I'm just thinking scientifically, not, of course, the regulatory and all that comes into play with that.
Well, theoretically, doxycycline might be sufficient, or another combination might be sufficient. I know there's another group in England that is doing a drug repurposing trial in London called the Care Oncology Clinic. And they're using a series of 4 drugs, one of which is doxycycline. And what they're finding is that it is actually very effective. They're all FDA-approved drugs. This is another trial of drug repurposing. And they did publish a paper already in Frontiers in Oncology, I believe, or another Frontiers journal. And what they showed is they could double survival in patients with glioblastoma.
Mm.
Glioblastoma is thought to be one of the universally lethal cancer types. It's a brain tumor.
Mm-hmm.
And with their therapy, which involves 4 FDA-approved drugs, which I believe all are in one way or another mitochondrial inhibitors, they could have a great effect. So on survival, which is the toughest thing, is to have an effect on survival. So I think there's promise here. We just need to get the government and regulatory agencies behind us in terms of testing these drugs. And that doesn't mean that you can't develop new drugs, because a lot of these drugs were not made for treating cancer. So you could also take these drugs and modify them to make them more effective. So it just provides a starting point. Instead of starting from 0, you could start from an FDA-approved drug and make it better.
And it also provides a financial incentive for the big drug companies, if they could be persuaded that this is a pathway worth exploring, to find those new drugs that they can develop and have that.
Exactly. So if you can show proof of principle with your FDA-approved drug, which is generic, then you've shown that there is value here, and then maybe the companies would be interested to help with your new trials. I'm sorry, I just want to.
Sure. No problem.
Sorry.
No problem.
All I'm saying is that FDA-approved drugs and drug repurposing is a very fruitful area, because number one, you can have something already safe, and you can move that from phase 1 to phase 2, because those drugs have already gone through phase 1, phase 2, and phase 3. So we already know they're safe and efficacious for one particular indication. So you can skip phase 1, which is a huge step, which costs a lot of money and saves a lot of time, and then bring those drugs right into phase 2, where you can look for efficacy, and then also you need to compare them to the standard of care. So that would be phase 3. So it just allows you more flexibility and costs less money.
So theoretically, again, we're just in the science principles of this. These antibiotics could eventually compete with the current standard of care if there was enough of a momentum behind it and resources to explore these things and to see how effective they could be.
I think that's one possibility, but more likely they would be used as an add-on to the standard of care to prevent
Yeah, I've just seen so many people
That's,
use the chemotherapy and everything and just devastating physically for the rest of their lives. So it's just, if you could skip such a toxic solution for so many people, if you catch these diseases early on, my goodness, it would be tremendous. If
that
you
would be
take,
the ideal situation
you
where
know,
you could use just an antibiotic.
but that's the terrifying thing, is if they're always going to make you have to go through toxic levels of chemotherapy, and then we'll give you your antibiotic, which is the
Well, you
thing that
might,
could have killed it from the beginning without all the hell, you know?
and you might be able to dial down the concentration of the chemo if you had another drug which was synergistic, because the toxic side effects, as you're probably well aware of, induce senescence and aging. So that's one of the problems of chemotherapy, is that if you've ever seen a cancer patient before and after they've taken the
Right.
chemo, it looks like they've aged 10 to 20 years, but that's because they did.
But
Because
it doesn't make sense under your principle that if senescent cells are the fertile ground for cancer, to turn on all the senescence you can with chemotherapy. You know what I
well,
mean?
that's part of the problem, is that if you ever look at the label on a lot of the anti-cancer drugs, you'll see that they're considered to be carcinogens. And when they're destroyed, they have to be incinerated because they're carcinogens. And a lot of patients actually develop secondary cancers. So you'll see, if you look in the literature, you'll see that I'm not saying we shouldn't do chemotherapy,
Right, right.
but I'm saying that there are side effects of chemotherapy, and one of them is secondary cancer. So a lot of patients after chemotherapy and radiation therapy, they develop leukemia and lymphoma. They don't do that overnight, but it could be 5 or 10 years, and there is a risk, and all of that is published. So if you could use less radiation and less chemotherapy, then potentially you could reduce those side effects. But it wouldn't be crazy to have another drug that you put in place to reduce the risk of secondary cancers or metastasis. And that could be the antibiotic. That could be the role that certain antibiotics like doxycycline could ultimately play as a prophylactic against recurrence, metastasis, and drug resistance.
I think if you're, again, we're at the scientific theoretical stage here, but if there is promise and it goes and continues where you're going with these antibiotics, I think the Hippocratically consistent way to approach human health would be to start with antibiotics that are not going to destroy the body as viciously as some of these other things. If you could catch these cancers at an early stage, you know, maybe I'm just thinking, I'm 31 years old. I'm thinking when I'm 70, maybe when we've had a few generations at the chance here, you know, 80, maybe somebody would have thought to try a more humane approach first before chopping and burning and destroying the whole body, you know.
I don't know if you read my article, but it was published in The Conversation a year or two ago when we discovered the energetic cancer stem cells. I wrote an article which was a lay article published in The Conversation, and I was very surprised. It was actually picked up by Newsweek, and it was republished by Newsweek. So I can send that to you, but in it I describe exactly what you're talking about. And we identified a new type of cancer stem cell that is very energetic. And we think that this could be the cell that's responsible for the recurrence, metastasis, and drug resistance. So obviously, you know, one part of our work is senescence, but another part is cancer. And because they're so inextricably linked, we feel that it's necessary to work on both aspects of senescence and cancer.
I wanted to quickly ask you along this line, has there been any scientific literature that some of the antibiotics that you're using have had any negative effects against cancer treatments and therapies? Have they, you know, confounded some of the traditional standards of care when used in any literature that you've seen? And then the second piece would be, I've heard azithromycin recently has gotten some heat because of potential heart condition issues with the rhythm of the heart. Have you
Right.
seen
With
that?
any antibiotic or any drug, there's always a risk of arrhythmias. Azithromycin has a risk, but it's still FDA approved, and obviously the FDA thought that that risk was not high enough to be a concern. Otherwise, they would have revoked its approval. So I think, you know, every drug has side effects. A lot of drugs have potential arrhythmia side effects. So, you know, everything is a kind of a question of risk-benefit relationship. So if you don't have to take a drug, that's better. But if you have to take a drug, it doesn't have to be all the time. We don't know how long you have to take it for its effect on senescent cells, but it could actually be quite quick. We've seen the effects are within 5 hours in cultured cells. So it's actually quite a rapid effect on the senescent cells. So we have to do further studies, but you know, people who are quite fragile, people who are patients with idiopathic pulmonary fibrosis, their survival in this trial that was published in Japan, their survival over the treatment, the conventional treatment, the survival was 25%, and with azithromycin, I think it was pushing 80%.
Wow.
So these are very fragile patients, and to go from survival of 25 to 80% is remarkable because of the reduction in the fibrosis. So, you know, as I said, everything is a risk-benefit. So all drugs have side effects.
Has
So
there been any literature that suggests that these antibiotics can, you know, mess with the standard of care protocol in terms of, you know, reducing the, you know, the effectiveness of the standard treatments?
there's only one treatment I'm aware of, and it was a very generic study where they didn't talk about, as far as I remember, the specific antibiotics, but they thought with immunotherapy that taking antibiotics was a risk. But we don't know if that's because those patients were already infected with bacteria. And so that's a confounding variable, because why were those patients taking antibiotics in the first place? And why were they giving them immunotherapy if they had an infection?
Right.
So we don't know if that was because of the antibiotic or because they were infected. And if they were infected, they shouldn't have been undergoing immunotherapy, because
Right.
you're, so I think that needs to be still worked out. Immunotherapy is obviously very promising, but I don't think any single therapy will cure cancer or aging. So I think we need to have a multi-pronged approach. And one therapy does not necessarily preclude another therapy or another avenue of investigation.
Right. That's fascinating. So what I want people to understand is that the first step, of course, is that we think in this immediate place that we're at, we need to spend more attention, public attention, and medical professional attention at this pathway that you've potentially identified here with the COVID-19. That's obviously the pressing issue that's on everyone's minds. It's could potentially destroy world economies if they can't figure this thing out and get this thing, you know, mitigated as best as we can. As you suggested, potentially a pathway with these antibiotics as a preventative, potentially, or as even as treatment too. So that's the first priority. Second priority is, of course, the long-term interest that we need to generate in the public square about the anti-cancer properties of these potential solutions here. So those are two huge things, you know. If we can use one, if we can kill two birds with one stone, with one antibiotic stone, that's a good thing, right? Just starting
You
there.
could save the world from the pandemic and also take care of aging and cancer at the same time with antibiotics. Why not?
That's the biggest story. That's like the two big, those are the two biggest stories of medical history in our time. If we could do that, if that could happen. So what do
And we
people,
have the patient populations to do these studies, so we should take advantage of that. And the drugs are cheap as dirt.
yeah, they're cheap as dirt, and we need. So, what would I mean? I know it's kind of putting you on the spot here, but what would your I mean, if you could recommend someone who is, you know, the medical professionals that are interested in doing a human trial on using these, you know, azithromycin or doxycycline, what would you do for? What would you recommend to folks? You know, to try one or try both up front. You know,
Well, obviously they already have something that works, so I guess they have to stick to that. But I think, you know, for trials in viral replication, you could pick another virus, and you could do the same thing with a virus that's not lethal,
Right.
and show that it inhibits viral replication. And then you could extend that to any of the other viral illnesses. So, but I think we do need trials. But for an emergency situation like this, like that doctor you were describing earlier in New York, he's prescribing off-label.
Right.
So it's at the discretion of the practicing physician. That's not as clear in Europe and the UK. There are tighter regulations. So I think the United States and maybe other countries are in a better position because there's more flexibility in their physician prescribing rights. So, but when I talk to oncologists, they don't think anything of prescribing doxycycline for a cancer patient because they know that their patients are more prone to infection, and they know doxycycline is a very well-established drug for over 50 years. That means that you know the behavior of the drug, you know the side effects, you know the pharmacokinetics, so how long it lasts in the body. So when you have a drug like that, which has stood the test of time in terms of 50 years of FDA approval, with minimal side effects, why not explore other, perhaps more profound uses besides acne, for example?
Well, for COVID-19, I was thinking that if folks could, I don't know, I guess I'm totally an outsider to this field, but I mean, taking people with mild symptoms who are younger, right? So they don't have to worry as much about that, and giving them some doxycycline, doing a human trial with younger, healthier patients that are having mild symptoms, and see if that makes an effect, maybe
That would actually be a brilliant idea. So because the standard of care that's been established, I guess, for treating patients is patients that are more emergent. So if you treated asymptomatic patients, there would be no ethical issue there, presumably.
Right.
So then you could just measure viral titers in the blood or in saliva or mucus, and you could establish that in these patients that are asymptomatic, you could establish whether the viral titers go down, which would be the gold standard now.
Right.
And the PCR tests, the DNA tests already exist, and there are new tests being developed also for antibody positivity. So all of those things could be done with young asymptomatic individuals, and you could quickly assess the relevance of doxycycline or azithromycin or other
or
drugs
even mild symptoms, right? Would mild symptoms, expressing even mild symptoms, would that still fall in that same category, or would they make it a little more tricky?
The patient wasn't severely symptomatic in a life-threatening way.
Right. So that way they can use the cocktail, right? Use the hydroxychloroquine and azithromycin and zinc or whatever else they want to use for the more intense cases, right? And then use that way we could do human trials with doxycycline alone or azithromycin alone or together for the younger, healthier patients with asymptomatic situations, right? That would be a great human trial to do right now.
I would be delighted to be involved in something like that as a consultant or as one of the people who puts the trial together.
Well, I mean, in your your family has connections with Italy, so there's a lot of people that need this right now around the world that they could use this information.
The whole world has stopped, basically. I mean, the whole world is under pressure, and economically and medically, and it's just shocking that we're in this situation. So
what's amazing is that
we need something.
What's amazing, what I love about learning is that it's always these systems thinkers like you that you're able to go outside of your specialist field and observe the patterns that you're seeing, right? So that you're able to not say, well, I'm only going to stick to aging and I'm not going to observe the information and the truth that I'm seeing as it relates to cancer or as it relates to COVID-19. You're able to follow the data wherever it goes. And that kind of systems thinking is much needed because that's how you actually change paradigms. It's not when you get into if you get that granular specialist thinking, sometimes that's the best place to get groupthink entrenched, where you can never see outside of your framework, and you're never able to move the ball forward because you're not seeing that broader picture, you know.
Yeah, I agree with you. I think my specialty has really been and I've been a scientist for over 30 years, is really to take things from one field that other people can't see and then apply them to another field. And then you wind up creating a whole new field because, people didn't think there was any relationship between those two things.
Right.
But because you're an outsider,
Yeah.
you can actually see clearer.
Yeah.
Because the people who are insiders are overwhelmed with all the details they already see, and they can't see the big picture.
Exactly. Well, that's why I found you. That's how I found you, because I asked Dr. Yu, I said, he has a unique theory of the atom. And I said, well, if your theory is correct, how would it inform the way you would approach, just personally, you yourself, right? How would you treat cancer on yourself if you could, theoretically, knowing what you know about how the atom works? And he said, I would use antibiotics. And I said, what are you talking about? It can't be antibiotic. What do you mean? And he said, I only use Z-Pak. Azithromycin's the only drug I use. It's the only drug my family ever uses. We avoid all drugs except for that whenever we get sick. And he said, well, how would that work? He said, well, you can explain it, Dr. Yu, if you want, but but you said it, that these antibiotics are charged and the cancer cells are charged, right? And they neutralize each other. Tell us a little bit about that.
Yeah, the bacteria or this virus, based on that shape. So I know this is a very similar to physics phenomena. So that's called a magnetic charged particle. So in order to cure magnetic charged particle, what do? You use the opposite field to neutralize them. So I said, well, antibiotics has a highly magnetic property. So so Dr. Lisanti might know this one. They use a magnetic field to direct antibiotics drug how to go. So that means antibiotics has that property too, right? When they are opposite field combined, they neutralize. So that's why they reduce their toxicity.
So what I did was, as soon as he said that, I went and started researching, and I said antibiotics and cancer, and I found your team. So here you go. You go from physics, gives me an intuition. Oh, wait a second. Let me see if this is true. Because Dr. Yu's theory, his physics perspectives have panned out very correctly on many other areas, more in line with traditional physics claims that we've investigated together. But when he made that, said he's too credible on everything else he said. When he says he would use antibiotics to treat cancer, in his mind, I said, I'm going to take that to the bank and see if I find something. And then I found your work. And he had never seen your work. So I just think it's interesting that the truth will always lead you in the same pathway. You know what I mean? It always
I think
goes.
you need to be open-minded, and you need to take other people's ideas into consideration. Even if they may seem ridiculous initially, it's obvious that you need to do experiments to test them. And so a lot of people may have a knee-jerk response where they may say, oh, that's garbage. But
yeah,
you need to reevaluate what are the facts and what are the details before you reject a new idea as crazy. You need to stop and think and see maybe there's validity there. So
another cancer biologist that we've interviewed a few times is Dr. Thomas Seyfried of Boston College. Do you know him?
I've heard of him, yeah.
Yeah, now he has a similar. He talks about its mitochondria is the problem in the cancer cell, and so he uses ketogenic therapy where he tries to starve the cancer cell of their fuel, the sugar, right? And then he uses some other treatments alongside of it. He uses a drug called DAWN, which is a glutamine inhibitor, to prevent the cancer cells from, using glutamine or sugar, and he starves it out that way. I mean, do you think that might have some compatibility with your approach?
Yeah, absolutely. I think any way you can to turn off the mitochondria would be potentially valuable. Mm-hmm. So there are a lot of different ways to turn off mitochondria. One is by targeting the mitochondrial biogenesis. Another is to target the mitochondrial energy production. And we just need to find which ways are the safest and which ways are the most effective through clinical trials. But you know, the starting point is the same, which is to identify mitochondria as the target.
Yeah.
And once you figured out that mitochondria are the target, then it's a question to figure out how to turn them on and off. And just to turn them on off in the cancer cells and not in normal body cells is the trick, and to reduce the side effects. And so therefore, you know, you could reduce the metastasis, the recurrence, and the drug resistance. So
I mean, I'm delighted at the dream, just speaking for myself, at the notion that one day people could be prescribed, doctors would know how to help people. This is my opinion, getting people on a therapeutic ketogenic situation where you're removing the sugar, so you're not feeding the fuel of cancer, and then you have that kind of monitoring situation, and you can use some antibiotics like you've suggested, and maybe some new ones that are created would be very cheap, and that people wouldn't have to be burned and cut up and chopped up and all that. To me, I see that picture and I want that for my children's generation. I don't want them to have to grow up in the terror and fear of cancer and its treatments that so many people have to deal with. So that's my personal goal, is to see people being treated therapeutic ketogenic situations and antibiotics as the front line of defense against these diseases, especially when they're caught early, you know?
right, and there's no reason why you couldn't use a glutamine inhibitor together with one of the antibiotics.
Wow.
To get a more synergistic effect to prevent the cells.
Are you familiar with Dawn, that glutamine inhibitor drug?
No, I'm not familiar with that one, but there are others that people have used, and you know, I think mitochondrial inhibitors will be the future for cancer therapy.
But both Dr. Seyfried's approach and yours does focus on the mitochondria, though, which is important. That's the paradigm shift that I think has to happen, right?
And also, berberine is a mitochondrial inhibitor, and it's been used by the Chinese for over 3,000 years as a traditional Chinese medicine.
Wow. Well, this is fascinating stuff. I really appreciate your time. I know we've gone a little long, and there's so many things to talk about with you, and we've had such a great time talking with you. Yeah. I hope you
meeting you both as well.
What was that?
It was great meeting you both as well, and we'll have to keep in touch.
Yeah, and I will do my part. This is just off the record. I will do my part to see what we can do to get the ball rolling for finding someone here in the States who can help facilitate a human trial. Because one of the things I do with my media program is it's an information clearinghouse where with my network of folks that I've been able to develop, I'm not just doing this as media. I'm doing this to get people on the ground, to get things done, especially when you have a pandemic situation like we're in. So. And by the way, do you know of anyone, I'm just going to ask you because I'm looking for them too. Do you know anyone in the States who has the kind of team that could do a human trial or something, if there's some funding or something to help get it going?
Yeah, no, I don't know off the top of my head because I'm not in the field of infectious disease and virology, but I think we should be able to find somebody who's a virologist. It seems like there's a lot of people now working on COVID-19.
Yeah.
You just have to find the most interested party to join us in this quest.
And they need to be a virologist. It can't be something outside of that field,
well, I
right?
think
To be credible.
Infectious disease specialist, but we could put together a team of people to do this.
Right. Well, I'm going to do my part, and I really appreciate what you've done. Dr. Yu, any last thoughts or comments?
One more question.
Okay.
Have you tried azithromycin yourself, CPAC yourself?
Well, once when I had a lung infection, I did take upper respiratory infection. I did take azithromycin, and I do feel better from having taken it. I don't know if it had any effect on the senescent cells, but I'm getting older now, so I'm about 56. So I thought it was interesting when I did take it for the upper respiratory infection. I did feel more with it, more conscious. So I did feel some of the positive effects, and that was after I had identified it as a senolytic. So, and I know other people who have had the same experience, where they felt a sort of clarity of thought that they hadn't experienced before.
Exactly. I took, a CPAC But since I get to move to Florida almost 17 years ago, and that's the only drug me and all my family members took. That's all needed. And I now, when I find out that this is the CPAC can treat, it's a one bird, four stones, not just cancer and anti-aging, and also antibacterial and the virus. So that's why I thought that this is a wonder drug to me.
It is. It's unbelievable.
Yeah. I'm curious to see what the relation is with its mechanisms and metformin, because they use that for anti-aging. Some of these folks are using that.
Well,
David Sinclair. Yeah.
That's another mitochondrial inhibitor. It inhibits complex 1 of the mitochondria.
Wow.
So there's a whole bunch of connections here, all revolving around mitochondria. So we should take the mitochondria more seriously.
That's where the future of medicine might lie, in that area, huh?
Mitochondrial medicine, yeah.
Well, so good to talk to you. It's been a pleasure. So we're going to keep in touch with you, and we could pick your brain for hours, as you can see, but
Okay.
we'll let you go. Thank you.
Thanks, guys.
Thanks.
Thank you.
Cheers.
Bye-bye.
Bye for now. Cheers. Bye.
Bye-bye.